Hello students, welcome to Rayat Online School Education Project, that is the ROSE project conducted by Rayat Shikshan Sanstha Satara, Sadguru Gadge Maharaj College Karad, conducted here 12th ROSE project 2021 to 2022. Myself, Miss Moite P.S. My subject is biology. My topic name is enhancement of food production. This is our fifth lecture of enhancement of food production. In this topic, we see microbes in energy generation. Here, many developing countries are encouraging for installation of biogas plants to meet out the requirement of energy. Biogas is used as a domestic as well as industrial fuel. Biogas is a mixture of methane, that is CH4. The methane is 50 to 60% methane present in the biogas, CO2 30 to 40% CO2, H2S, that is the 0 to 3%, and other gases are CO, N2 and H2, carbon dioxide, nitrogen and hydrogen in traces. Biogas is highly inflammable and is used as a source of energy. Plant waste, animal waste, domestic waste, agriculture waste, municipal waste, forestry waste, etc. are commonly used for biogas production. Cattle dung is most commonly employed substrate for biogas production. It is a rich source of cellulose from plants. Go to next slide. Here we see biogas production. Most commonly used models of biogas plants are KVIC and IARI. The digesters used for biogas production is called a biogas plant. A typical biogas plant using cattle dung as a raw material consists of digester and gas holder. Digester is made up of concrete bricks and cement or steel. There is a cylindrical gas holder or gas tank above it to collect gases. Digester has a side opening, that is the charge pit, into which raw material as cow dung is fed. The digester is partly buried in the soil. Go to next slide. We see here biogas plant. Here dung water is called, this part is called as a charge pit. Here gas holder, and here this biogas is produced. This tank is called a digester, and here sludge. This is the biogas plant. Go to next slide. Anaerobic digestion involves in three processes. First process is hydrolysis or solubilization. In initial stage, the raw material is used, cattle dung, is mixed with water in equal proportion to make slurry, which is then fed into the digester. Here, anaerobic hydrolytic bacteria, that is example Clostridium, Pseudomonas, hydrolyze carbohydrates into simple sugars, proteins into amino acids, and lipids into fatty acids. Go to next slide. Acidogenesis. In this stage, facultative anaerobic acidogenic bacteria and obligate anaerobic organisms convert simple organic material into acids like formic acid, acetic acid, hydrogen, and carbon dioxide. Methanogenesis. In this stage, this is the last stage in which anaerobic methanogenic bacteria like methanobacterium, methanococcus convert acetate, H2 and CO2, that is hydrogen and carbon dioxide, into methane, carbon dioxide and H2O and other products. Go to next slide. Benefits of a biogas. See here, it is a cheap, safe and renewable source of energy. It can be easily generated, stored and transported. Second benefit is it can be used for domestic lighting, cooking, street lighting, as well as small scale industries. Third benefit is it is burns with blue flame and without smoke. Fourth benefit of biogas is it helps to improve sanitation of the surrounding. Fifth benefit is it is eco-friendly and does not cause pollution and imbalance of the environment. Sludge which is left over is used as a fertilizer. Go to next slide. Here, role of microbes as a biocontrol agents. In this point we see the term biocontrol refers to the use of biological methods to control diseases and pests. The natural method of eliminating and controlling insects, pests, and other disease-causing agents is by using their natural biological enemies. This is called biocontrol or biological control. The agents which are employed for this are called biocontrol agents. Microbes are one among them. Go to next slide. Microbes are one among them. These microbes include bacteria, fungi, viruses, and protozoans. Microbes as biocontrol agents act in three ways. Either they cause the disease to the pest or complete or kill them. Chemicals, insecticides and pesticides are extremely harmful to human beings and also pollute our environment. Hence, the use of biocontrol measures will greatly reduce our dependence on toxic chemicals and pesticides. Go to next slide. Example of a microbial biocontrol. That is Bacillus thuringiensis, that is a BT, is used to get rid of a butterfly, caterpillar, where dried spores of a Bacillus thuringiensis are mixed with water and sprayed onto vulnerable plants such as brassicas and fruit trees. These spores are then eaten by the insect larvae. In the gut of the larvae, the toxin, that is cryprotein, is released and the larvae get killed eventually. Go to next slide. Trichoderma species are free-living fungi found in the root ecosystem, that is the rhizosphere. These are effective as a biocontrol agent of several soil-borne fungal plant pathogens. The fungus produces substances like viridin, gliotoxin, gliovirin, etc. that inhibit the other soil-borne pathogens attacking root, rhizomes, etc., causing rot disease. Go to next slide. Here, microbial pesticides and their host. The correlation is depicted as per the following table. Here we see pathogens and their host. Here, bacteria Bacillus thuringiensis, that is a BT. Their host is a caterpillars. Bacteria Papilli, their host is a cabbage worm, and bacteria Lentimorphus, that is adult beetle. There is the in these three pathogens, their hosts are caterpillar, cabbage worm, adult beetle, etc. Fungi. Here names of fungi: Beauveria bassina, Entomophothora, Pallidaroseum, and Zupthora radicans are found in host aphid croci, a unigloculata, mealybugs, mites, white flies, protozoans. Here, Nosema lacusti are found in grasshopper, caterpillars, and crickets. Viruses, that is the nucleopolyhedrovirus, NPV, and granulovirus, GV, found in caterpillars and gypsy moth, ants, wasp, and beetles. This is the microbial pesticides and their host, that is the pathogens and their host. Go to next slide. Here we see bioherbicides. Weeds are the unwanted plants that grow in agricultural fields, ponds, lakes, etc. Weeds compete with the main crop in the farm land for water, space, minerals, light, air, etc. and also act as a collateral hosts for several pathogens. Microbes are also used as herbicides. Many dicot herbs that grow in the field of cereals as weeds can be killed by certain microbes. Go to next slide. Here we see microbial herbicides and their host. First, that is the pathogenic fungi as a mycoherbicides. Fungi name is a Phytophthora palmivora, controls milkweed in orchards. Alternaria crassa controls water hyacinth. Fusarium species controls most of the weeds. This is the pathogenic fungi. Bacterial pathogens as herbicides. Pseudomonas species attacks several weeds. Xanthomonas species attacks several weeds. Agrobacterium species attacks several weeds. Third, this is the bacterial pathogens. Here, herbicides as herbicides. Here, insects as herbicides. In insect, Tyria moth controls the weed Senecio jacobi. Second insect, Cactoblastis cactorum controls cacti. Go to next slide. Here we see in this slide role of microbes as a biofertilizers. Fertilizers are nutrients which are necessary for the growth of plants and thus for the productivity of cultivated plants. Use of fertilizers for increasing productivity is one of the aspects of a green revolution. Fertilizers are classified as inorganic, that is the chemical, and organic, that is the biological. Inorganic fertilizers are synthetic, where mineral salts of NPK are mixed in a definite proportion and then dusted in the field. Excessive use of such fertilizers lead to pollution of soil, air and groundwater. Go to next slide. Organic fertilizers are biological in origin and include farmyard manure, that is the FYM, compost and green manure. Use of these fertilizers increases the fertility of soil. Nowadays, for better the sustainable agricultural production, farmers use biofertilizers and practice organic farming. Biofertilizers are mostly N2 fixing, that is the nitrogen fixing, living microorganisms which enrich the nutrient quality of soil. They include bacteria, cyanobacteria and fungi. Go to next slide. Biofertilizers are commercial preparation of ready to use live bacterial or fungal formulations. Their application to plant, soil or composting pits helps to enrich the soil fertility due to their biological activity. Use of biofertilizers is cost effective and eco-friendly. They play a vital role in maintaining a long-term soil fertility and sustainability. Go to next slide. Types of biofertilizers. Biofertilizers are divided into following groups. First is N2 fixing biofertilizers. Here, the nitrogen fixing microorganisms which convert atmospheric nitrogen into nitrogenous compounds like nitrites and nitrates via ammonia. Symbiotic N2 fixing microorganisms, for example, Rhizobium, Anabina, Frankia. These are always associated generally with underground parts, that is a root of higher plants. Next, see here next, free living or non-symbiotic N2 fixing microorganisms, that is example Azotobacter, Nostoc, Clostridium, Bergeringia, Lepsilla, etc. Go to next slide. Phosphate solubilizing biofertilizers. These are the bacterial species which solubilize the insoluble inorganic phosphate compound such as rock phosphate. For example, Pseudomonas striata, Bacillus polymexa, Agrobacterium, Micrococcus, Aspergillus species, etc. Compost making biofertilizer. Composting is a natural process that turns organic material into a dark rich substance called as a compost or a humus. The composting process is dependent on microorganisms to break down organic matter into compost. There are many types of microorganisms found in active compost, such as bacteria, fungi, actinobacteria, protozoa, and rotifers. Go to next slide. Cyanobacteria as biofertilizers. Many cyanobacteria are aquatic and terrestrial, free-living or symbiotic, aerobic, photosynthetic, N2-fixing, heterocystous or non-heterocystous forms. Example: Anabina, nostoc, Plectonema, oscillatoria, etc. Anabina, nostoc, and Tolipteryx are associated with lichens, while Anabina is associated with plants like Azolla and Cycas. Here, fungal biofertilizer. Next point is a fungal biofertilizer. Mycorrhiza is a fungus. It forms symbiotic association with the underground part like rhizomes and root of higher plants occurring in thick humid forest. These are discovered by Frank in 1885. There are two types, wizard ectomycorrhizae and endomycorrhizae. Ectomycorrhizae, they have well-developed mycelium that forms mantle on the outside of the roots. Due to this, the plant vigor, growth and yield increase. Some hyphae of mycorrhizal fungus penetrate into the root and forms hiding net in the intercellular spaces of the root cortex. Go to next slide. Endomycorrhizae, they grow in between and within the cortical cells of roots. Fungal hyphae penetrate the cells and form finely branched arbuscules intracellularly and form vesicles mostly in the intercellular spaces of cortical cells. Hence, they are called vesiculo-arbuscular mycorrhizae or a VAM, short form of a vesiculo-arbuscular mycorrhizae, that is the VAM. The plants with VAM grow luxuriantly in less irrigated lands. The association of VAM with crop plants helps in a conversion of a less productive field into more productive field. Go to next slide. Biofertilizers, microorganisms. First microorganism is a rhizobium. Rhizobia are rod-shaped, motile, aerobic, gram-negative, non-spore-forming, nitrogen-fixing bacteria containing node genes and NIF genes. They form symbiotic association with roots of leguminous plants. They bring about nodule formation on the root and multiply inside the nodule. They fix atmospheric nitrogen into organic forms which can be used by plants as nutrients. For example, R. leguminosarum is specific to pea plant, and R. fascioli to beans. Go to next slide. Here we see root system of a leguminous plant. In this figure we see root branches, and on the root branches root nodules are present. Here next diagram is the TS of root nodules. Here we see TS of root nodules. We see here phloem, xylem, this is the root, and here nodules on the This is the root nodules in which bacterial colonies in a cortical cells. Go to next slide. Azotobacter. It is the important and well-known free-living nitrogen-fixing aerobic, non-photosynthetic, non-nodule forming bacterium intimately associated with roots of grasses and certain plants. It is used as a biofertilizer for all non-leguminous plants, especially rice, cotton, vegetables, etc. Azos, Azospirillum, third Azospirillum. It is a free-living aerobic nitrogen-fixing bacterium associated with roots of corn, wheat, and jowar. It fixes the considerable quantity of nitrogen, that is the 20 to 40 kg, in non-leguminous plants such as cereal, millet, cotton, oilseed, etc. Next bacteria is Anabina. It is a genus of multicellular filamentous cyanobacteria that exist as a planktons. It has ability to fix nitrogen and also forms symbiotic relationships with certain plants such as the coralloid roots of Cycas and Anthoceros thallus. It has some specialized and colorless cells called heterocysts, which are the sites for nitrogen fixation. Go to next slide. Here we see next bacteria, that is the Azolla. Azolla is a free-floating water fern. Sorry, this is not bacteria, this is water fern. Azolla plant consists of a floating rhizome, that is a stem, which small overlapping bilobed leaves and roots. The leaf shows dorsal and ventral lobe. In the dorsal lobe, Anabina filaments are present in the parenchyma, which fixes nitrogen. Azolla can be used as a biofertilizers in the rice field. Here we see Azolla plant. LS of Azolla leaf showing filamentous Anabina. Here, Anabina present here. In the longitudinal section, here we see cavity. This is the dorsal lobe. Photosynthetic zone see here, and here Anabina is present. Benefits of biofertilizers. First benefit is low cost and can be used by marginal farmers. Second benefit is free from pollution hazards. Third benefit is increased soil fertility. Fourth is BGA as a biofertilizers secretes growth promoting substance, organic acids, proteins, and vitamins. Azotobacter supply nitrogen and antibiotics in the soil. Sixth benefit is biofertilizers increase physicochemical properties of soil like texture, structure, pH, water holding capacity of soil by providing nutrients and organic matter. Here, our enhancement of food production topic is complete. Thank you.